All right, welcome everyone to our seminar today. It's my pleasure to introduce Eric Pedersen. Eric is a community ecologist, a theoretical ecologist, and a fisheries biologist, now at Concordia University, where he is an assistant professor. He studied at the University of Saskatchewan, actually with biology and a minor in economics, which I found really interesting, and then did his PhD at McGill. Moved on for a couple of postdocs, worked at the Department of Fisheries and Oceans for a while in Newfoundland, so in our region here. And he's now coming to us from Ottawa. And I'm really happy that we have Eric here today talking about regime shifts in diverse communities. If I can ask everyone to please keep muted during his talk. And you can always ask questions through the chat. We'll probably answer them in sequence afterwards. But as you think about questions, maybe you can write them down so we have a log of questions and can launch into a question period after the talk. So Eric, thanks again for joining us today and for your talk.
Thanks, Boris. And thank you everyone for being here and for the kind invitation. It's really exciting to be back in Halifax, even if virtually. So my group works on a whole range of things. We focus on a mixture of theoretical ecology, so trying to model and understand rapid change in ecological communities, statistical ecology, so trying to develop new methods to try to detect change in ecological systems, especially in diverse and complex ecological systems, and applied fisheries ecology. So trying to understand causes and consequences of rapid change in productivity and how it affects long-term fisheries models. So today I'm wearing my statistician hat. I'll be focusing on a lot of the work I've done the last few years on trying to detect regime shifts, so trying to detect abrupt and rapid change in ecological communities. We've known for a long time in the history of population ecology that populations, when they fluctuate, when they change, often follow predictable patterns. So this is a plot of walleye, which is a freshwater predator species in Escanaba Lake, sampled from 1990 until roughly 2012. And this is just showing a portion of this time series for this fish population. But the whole dynamics for the population going back to the 1950s show a pretty consistent pattern. You see a population that fluctuates around some average value, going up and down, but generally tending to show this return to a common trend. So this is the basic idea of density dependence, that populations will return back to some long-term average after they're perturbed away from it. And of course, we see more complicated dynamics in some systems. So this is a time series of lynx that have been caught by hunters from the 1800s into the early 1900s. And what you see is a population that cycles up and down, but if you take each one of these cycles, so you just take one of these cycles and transposed it from one point to the next, what you'd see is a very predictable pattern. You'd see the cycle is repeating itself. Even though this population is more complex, so it's fluctuating in a more complicated way, it's inherently still following a predictable pattern of repeating cycles. However, as we studied communities and we studied ecological populations, we've started to observe more and more cases of rapid, unanticipated, and unpredictable change. So this is actually from an experimental study done by Steve Carpenter and colleagues in northern Michigan, looking at eutrophication in a single lake. So in this lake, they had essentially initially a low population of largemouth bass, which are predator species, and low phosphorus. So this was a lake that had not much nutrients added to it. It wasn't heavily eutrophied. What they did is they divided the lake in half and perturbed both sides. So in both sides, they added nutrients, and on one side, they added bass. So they added essentially a large predator. And what they were able to see is that in both cases, adding nutrients led to increased productivity. But on the left side, where there was no predators added, they saw this massive increase in phytoplankton. So they saw obviously a eutrophication or an algal bloom. On the right side, they saw no such change, but they actually saw the arisal of a consistent and persistent population of bass. So this bass species that was able to eat small fish which in turn let zooplankton flourish, which in turn kept phytoplankton down, was able to persist long after the researchers stopped adding bass to the system. So we see a system that's in fact a single lake and divided only by one curtain has formed into this persistent long-term split into two different regimes or two different ecological systems. So we see this not only in experimental systems and not only at small scales. So these are time series of catch per unit hour of northern shrimp. So this is a northern cold water shrimp species that I study quite a bit. And what we saw in Newfoundland in 3 different channels in the 1980s was catch was quite low. So these species were low abundance. There's a commercial fishery, but it wasn't massive. Before in 1990s, we saw a massive rapid increase in the population, an explosion of almost 3 to 4 to maybe 10 times population size. And then we start to see again from 2008 onwards, this population has again started declining, except in the northernmost channels in the Hopedale region. We also see this occurring not only large populations, but across landscapes. This is from a study of walleye studied across lakes throughout Wisconsin. What we see is that across years, walleye density has been steadily declining. While this might look like a consistent linear trend downwards, this is actually plotted on a log scale. So this change is actually quite rapid, declining from 4 walleye per hectare in some lakes on average to an average about 1.5 to 1 walleye per hectare in a span of only 20 years. The population has essentially dropped in half across all these lakes and across this complex ecosystem in a very short period of time. And of course, there's one of the most iconic cases of rapid population change, which is the collapse of the northern cod. So this is a time series of fish landings of northern cod collected from 1800s up until the 1900s. And we see, of course, the rapid increase in catch from the expansion of the offshore fleet, the closure of that catch due to partially population collapse and partially closure of the Canadian Economic Zone to foreign fishing. But then in 1990, we see a population that goes from a relatively healthy state to all of a sudden plummeting to near to incredibly low levels, so 1% of its previous level. And we see this population that even after fishing stopped, it tended to persist in this very low state. This has led to the concept of a regime shift. So previously, we thought about populations as being in relatively persistent states where they would tend to return to an average behavior, whether it be cycles or a mean or even, say, chaotic fluctuations. But instead, we're starting to see increasing cases of these regime shifts. We see abrupt transitions between ecological states that persist over time. So even after we've stopped doing whatever we thought drove the system to that initial state, this new ecological state can persist over long periods of time. No, arbitrarily long lengths. So what I'm going to talk about today is how do we detect these shifts as they happen? There's been a massive amount of research on what causes them, the theory behind them. But what I want to focus on is just the fundamental question of how do we detect an abrupt shift as it happens? And how does it vary in systems of more complex than just single species? If you're all familiar with the concept of a regime shift, you've probably seen a diagram that looks like this. So this is a diagram showing some ecosystem state. And so the ecosystem state shown on the y-axis. So this could be mean population size. This could be, the abundance of the most common species. This could be, say, average body size, any sort of single indicator of the ecological state. And this system is essentially transitioning from a stable state, so solid lines here denote stable states, and as some ecological driver shifts, this could be, say, warming, or in the case of eutrophication, it could be a case of increasing phosphorus, or it could be increasing fishing pressure. This system doesn't change substantially, so it does go through maybe some decline of ecological state until reaching some threshold point here where essentially internal forces in the system are no longer strong enough to keep it at that previous equilibrium. And when a system crosses this point, it will drop to some new equilibrium point, which, depending on the theoretical system, like depending on the structure of the model used for the system, it might actually not be able to return to its previous ecological state, even if we relax the driver back. So going back to the cod example, this might be a case of cod populations staying quite high under increasing fishing loads and increasing environmental change, until at some point they drop off a cliff. And then even with the cessation of fishing, or the substantial reduction in fishing, they're not able to return for long periods of time. And so this previous diagram is showing what this looks like as essentially a theoretical construct. So if we could control the ecological driver and shift it at whatever rate we wanted, we could manipulate ecological drivers at large scales experimentally, we'd be able to get a pretty good reconstruction of this system. You know, we wouldn't be able to get this dash line, which would actually be an unstable state, which pushes it away, but we'd be able to get a pretty good picture of how this varies. But instead, we have to contend with time series data. What we have to contend with is trying to detect this abrupt change based just on variation in the population over time. And as an animation, what this would look like over time, if we imagine a persistent ecological driver, with this ball represents the current state of the population, what we'd see is it will go down and rapidly drop. So we think we can detect these abrupt changes or regime shifts as essentially a population that goes along, that rapidly changes at some point to a new level, then continues forward. The problem with this is that this is a very single population or single state based view of the population. We might not always be able to identify a single species or single organism that's actually able to track a regime shift in real time. So we know that populations are embedded in communities. In this case, for instance, this is the classic figure of the mess of the food web of Northwest Atlantic. And we see that both cod and shrimp are embedded in this food web and are interacting with each other. So we're looking at the rapid increase in shrimp or the rapid decline in cod. We can't think of these as two separate regime shifts. We instead have to think of them as multiple species coupled in a single regime shift. When we talk about a regime, we typically think about a regime as something that's encompassing essentially a whole community, or at least a discernible grouping of a community. So looking at a single species over time might not actually reveal the full picture of how a regime shift plays out. We also know that populations are embedded in landscapes. So this experimental lake, Long Lake, is only one of a matrix of lakes going across this whole landscape. And regime shifts aren't going to necessarily happen at all lakes or all locations in space at the same time or the same way. So if we want to try to quantify how regime shifts play out over spatially structured systems, we also need methods that are able to detect how the timing of regime shifts varies from one place to another. The question I want to focus on today then is, how do we detect rapid change across a large number of groups, where it be species or it be locations across time at once? So how do we actually quantify when a regime shift has started and stopped in complex and spatially and biologically diverse communities? Just as a brief summary of what I'm going to be talking about today, first I'll be talking about the problem of measuring rapid change in complex communities. Then I'll be talking about an approach we've developed called hierarchical generalized additive models as a method that lets us measure community rates of change at one time and create summaries to determine when the whole community is shifting. And then finally, we're looking at a couple examples of how we can apply this method of HGAMs to try to understand the patterns and causes of decline in complex systems. As I was saying before, we know that different species can respond at very different rates during periods of abrupt change. So this picture here is our classic view of what an abrupt shift or regime shift looks like. Over time, the species abundance starts high, might start declining a little bit as the driver starts impacting it, then at some point goes through a rapid change to decline. So this seems like something that should be easy to pick up. It seems like a pattern that should be easy to detect using some sort of statistics for breakpoints or change horns, or even just like fitting a linear regression on either side of this. However, in more diverse communities, these patterns of a single species rapidly changing don't necessarily always play out that same way. So for instance, you might have a case where the single most abundant species of the community stays very stable up until some point and rapidly plummets, but other more sensitive species were actually changing prior to that. So this, low abundant species might have been changing rapidly well before that. So we want to determine when this regime shift actually started. So when the community switched from one regime to another, the regime shift is actually starting here and only completing after all these species have reached a new equilibrium. So if we focus on just the most abundant species of the community, we might get a false picture of when a regime shift started if that species is not also the most sensitive. Further, we might also get patterns of shifting community composition during the regime shift. So here we have a case where one species goes from abundant to rare, and a second species goes from abundant to rare to common, and a third species stays at a stationary level. So in this case, just focusing on any one of these species doesn't give us a full picture of it. And if we try to focus on something like total biomass of the system, total abundance, we'd actually think that essentially nothing has happened here. The total abundance system has stayed essentially the same, and we miss the compositional shift. So to illustrate this issue of trying to detect rapid change in complex systems, I want to focus on the case of spatial temporal community dynamics on the Newfoundland shelf. So how did the whole community shift in Newfoundland? This is based on data taken from DFO from their standardized Charles survey from 1981 till 2013. This is a depth stratified survey of 141 depth strata, ranging in depth from 65 to 1,400 meters across this roughly 250,000 square kilometer region of the Newfoundland and Labrador shelves. And we focused on, from this trawl species, biomass of 29 groundfish species. So the abundances across space and time of these 29 species to see how they are shifting together. So this is from a paper we published in Royal Society Open Science in 2017, trying to dissect the community anatomy of this collapse. And what we see from this is that cod itself collapsed in 1990. So this is a species that was fluctuating around quite a bit prior to 1990. But then in 1990, we see this classic rapid decline of the species. And we see what might be first signs of the species starting to increase again only around 2010. However, if we look at all of the species of the community, or at least the 29 most common species that we focus on that were well sampled throughout the survey period, what we see is that the majority of these species actually started declining well before cod did. So this community, at least the abundance of this community, was essentially in free fall from roughly 1983, 1984 up until 1990, and didn't stop changing until 1995. So we look at just total biomass of the community, we can see this period of free fall pretty clearly. We can see that the biomass of the community was rapidly declining from this 1983 all the way to 1991. So well before what we think of as the start of the cod collapse, this community was rapidly shifting. And in fact, in some ways, cod was one of the last stocks to go through a substantial decline. What we also saw was that the community composition, so the makeup of the relative abundance of different species, was also shifting, but at a different rate than total abundance. So in the blue line here, we see what the community composition is. Looking at this as using an NMDS plot. So this is a basic ordination plot where points that are close together in this two-dimensional space are close together in community composition. What we see is from the 1980s up until 1990, community is staying relatively stable. It's not shifting rapidly. Then from 1990 until 1995, we see a community that's essentially rapidly shifting in composition. So this is after the major decline in biomass has occurred, the community has started shifting rapidly in composition. And then it's not until later we start to see a community that's returning, with this dashed line actually representing a gear change in the survey. So this dashed line indicating a point when the observation process itself shifted. But you can see that the gear change did lead to a relatively large jump in community composition as different species were being caught, but we're still seeing a substantial return to the initial community composition. So we're essentially seeing two distinct regimes here of this 1981 to 1990 period, and this second period of shifted community composition and possible slow return. And in case you're wondering if this is just being driven by shifting patterns in fishing pressure or shifting patterns in commercial catch, if we focus on just the 25 species that were not caught abundantly or not major commercial species or not frequent targets of commercial fishing activity, we see almost the exact same pattern of relatively slow change in the 1980s, rapid change in the 1990s, and then slow return. Although we see a bigger observation effect from the gear shift, partially because a lot of these species are smaller and were more easily targeted by the smaller mesh gear that was used after the gear change. So if we look at these as a whole bunch of different metrics and try to quantify when did this regime shift happen, what we see is that no single metric is giving us a complete picture of the collapse or recovery. So this is plotting 3 different metrics, so just community biomass in white, cod biomass in red, and community composition in blue. And what we can see is essentially both biomasses were declining rapidly. Community composition didn't start transitioning until the 1990s. And then we see different rates of return back to the previous state. And basically all 3 of these indicators have been scaled so that 1981 corresponds to 100%. And the largest difference we see in this metric from 1981 is scaled to 0. What we can see is that community composition actually started returning more quickly. So the community actually started resembling the initial community more rapidly than biomass did. So biomass here is essentially acting like a slow variable. So we try to determine whether or not the system is going through regime shifts. Actually, in this case, we need to look at all 3 metrics. And if we want to characterize when the regime shift actually occurred, probably our best estimate is it was occurring from roughly 1983 until 1995. So almost a decade-long period of change, instead of this rapid decline that we thought previously had thought of it as in the 1990s. So this raises the question then of how do we quantify and detect regime shifts in complex communities? If we can't look at any single metric to try and determine has it or has it not gone through a regime shift, we still need to find ways of detecting regime shifts. We can't empirically, or when we're trying to manage or monitor a system, try to track every possible variation or every possible system, part of that system to determine if it's changing rapidly or not at a given point in time. We still need metrics to quantify, is the system changing rapidly right now? So I want to ask the question of how do we actually detect regime shifts in complex communities? So what is a regime shift broadly? So I want to ask the question of how do we detect a regime shift? We have to ask the question what a regime shift is. And the simple answer to that is it's a rapid change in ecological state. So I mean, obviously this is just the definition I gave before, but it's emphasizing a different point. It's not emphasizing the idea of the state, it's emphasizing the idea of rapid change. So if a regime shift is a period of rapid change, why do we focus when we're trying to detect them on trying to find breaks in state or breaks in abundance? Why don't we actually try and focus on quantifying how quickly systems change at different points in time? So if we switch from this perspective of trying to quantify regime shifts based on abundance to trying to quantify regime shifts based on rates of change, we actually come up with a relatively easy metric of what a regime shift is. So again, if we get back to this picture of abundance regimes versus composition regimes, we can see an abundance regime is a period where average rates of change are low or 0. So across all species in the community, so here we have 2 different species, the abundance regime shift is one where, abundance regime, sorry, is one where the average rates of change are not changing quickly. So if we look at a bunch of different species and look at the average rates of change, so the slopes of all these different species averaged across them, we can see that in abundance regimes, we'd expect to see the average rates of change to be flat. And then in periods of regime shift, when everything is declining, we expect to see average rates of change to be low. Or if everything's increasing, we expect to see average rates of change to be high. We can pick out these regime shifts then as periods when rates of change differ substantially from the baseline. In the same way, we can also define the idea of a compositional rate of change or a compositional regime. And a compositional regime is just a period when variation in rates of change is low. It actually turns out if you look at variation in per capita rates of change, so how quickly different species are changing on a per unit biomass level, if the average composition of a community is not shifting, then the standard deviation of their rates of change should be 0. So we can actually measure how variable the community is shifting by looking at this standard deviation or variance in per capita rates of change. And periods of rapid compositional change, so on the top here, where we're seeing a bunch of different species all declining or increasing, we can look and see a period of high variance in rates of change. And we can identify when the regime started, a shift started and ended by periods where the variance increased and decreased. So that's a great idea. It's a useful thought that we might be able to measure rates of change. But can we actually identify regime shifts from these summaries of rates of change? Does this work in practice? And how do we go about measuring rates of change? So rate of change is essentially something we can never observe in the wild. All we can see is biomasses at different time points or abundances at different time points. And from that, we have to infer what the rates of change are. So what this part of the talk is focusing on then is trying to use a method we called hierarchical generalized additive models. And I'll explain what that is, to try to estimate rates of change across communities. So how do we estimate rates of change? If all we can measure are abundances or biomasses at different times, how do we estimate how quickly the population is changing? We have to deal with two issues when we're, at least two issues when we're trying to estimate rates of change across communities. The first is that real time series are noisy. So if we try to estimate rates of change as just the point-to-point difference in survey abundance, we'd have to contend with the fact that time series, or real time series, involve measurement error, they involve process error, they involve large fluctuations in populations from one time point to the next that are not necessarily associated with any sort of regime shift or abrupt change. So we want to try to find cases where the system is changing rapidly that average over these fluctuations. And the solution to this is that we can essentially fit a smooth trend to this data, basically something that penalizes small fluctuations towards an average trend. So treats those essentially as noise or process error, but then is able to smoothly and rapidly change when conditions are showing this period of rapid change. So essentially we're smoothing over these small fluctuations while still allowing the trend itself to vary non-linearly. And from this, we can actually estimate rates of change at each point in time. The second point we have to contend with is that when we're trying to measure rates of change for a large number of species at once, we have to deal with the fact that rare species are hard to estimate. So this problem of high fluctuations is even more accented in rare species where we might miss them in a single year, we might see large scale variation. We expect to see essentially more fluctuations in biomass in rare species when we're observing them in common species. But they still hold a lot of information. And as we saw before, rare species might actually be some of the most sensitive to rapid change. And we want to use them as bellwethers to help us detect rapid change. So how do we include them in our analyses? And the solution we're using here is essentially to try to borrow information on trends of rare species based on common species in the series. So rather than fitting a separate smooth curve for each species, we'll actually penalize these curves towards each other. So we put a penalty on how much these curves can vary from one another. So essentially pulling rare species towards their more common brethren. So still allowing estimated changes that differ in trend from the main or mean species, but still borrowing information. So if all we have for a rare species is just random fluctuations, the model will smooth over that and treat it as having essentially the same trend as more abundant or more common species. And the method we use to do this are what are called generalized additive models. So generalized additive models are a method that essentially lets us fit use the machinery of linear regression to fit smooth models with penalties on them. So lets us fit essentially a smooth curve to this data by basically breaking this X data into a bunch of different small functions, adding those functions up to create essentially a regression plot, and then penalizing those small functions towards each other to fit smooth curves. So essentially saying that there's a higher, I guess in a Bayesian sense, there's a higher probability of seeing a very smooth curve than of your trend, and then seeing a very highly fluctuating curve of your trend. And what we showed in a recent 2019 paper, essentially a summary and a tutorial, this concept of hierarchical generalized additive models is that you can actually move one level up from this. So rather than having a single curve that's penalized to be smooth, you can actually penalize multiple different species towards either a common trend or assume that they have a common level of smoothness. So you can essentially assume that different species should show similar levels of fluctuations, effectively borrowing information on the variation in trends from one species to the next. And to show how this process works with the Newfoundland and Labrador data, we can see how we'd apply this in one step at a time. So this is the biomass time series from Newfoundland and Labrador for these 29 key species. This is what happens when we smooth them. So these are all essentially penalized towards each other. So they have similar levels of smoothness. So now you're able to see the common trends from these. And what we can do now with these smooth curves is estimate rates of change. So how quickly each species is changing at a given point in time. And then from that rates of change, we can add them up vertically to find the average rates of change at a given point in time. So how quickly the whole community is shifting. And what I'm showing you is just one plot from a paper we just published in IC's Journal of Marine Science last year. What this does is gives us a clear picture of how the community as a whole is shifting. What this plot is showing is one of three. I'm just going to focus on this one and focusing just on abundance transitions because we're able to quantify community transitions in community state, but it actually showed very similar patterns. Was that essentially the community as a whole showed negative rates of change from at least early 1980s, so probably 1981 to 1982, showing rapidly declining rates of change up until 1990. So essentially the community was declining and declining much faster until 1990. And then we saw a period where the community as a whole, when measured as per capita rates of change, was slowly, was declining at a lower rate up until roughly 1995, 1996. And then we actually saw a period of actually slow per capita population growth before seeing another decline following roughly 2010 or 2012. So one of the interesting things about this is this actually highlights that what we thought was the regime shift happening in 1990 took place over a much longer period of time. So it took place from probably 1981, 1982, all the way up until at least 1995. So essentially a period when it's switched from one state to another, you can view it with the confidence interval. Wherever the confidence intervals overlap 0, it's a period where the model is saying that there's no substantial change in abundance or biomass. We're also seeing that the system never actually stabilized around a new equilibrium. So there's a period essentially following 1995 where the population is increasing again, and then following another period of where populations are on average declining again. So at no point do we see the system actually seeming to stabilize into a new, potentially stable regime. And in fact, as we're digging into this and looking at this, we actually think this might be a case of a longer transient, that the collapse of cod and the collapse of other groundfish might not have been a new stable state at all. It might have just been a period where it stayed at a very, very low abundance for very, essentially arbitrarily long periods of time. But eventually, if we let the system go and stay there long enough, it might actually have returned to its previous abundance, or might eventually return to its previous abundance. And these GAMs we fit were not just time series models, they were spatial temporal models. And the useful part about this is that we can actually use the same HGAM framework to determine not only when did a regime shift start, but where it started. So this is a map of all the depth strata used in the survey, as well as a map of where you see the deepest areas. So you'll see some deep areas of some of the channels, as well as the sharp shelf break. And of course, we have Newfoundland and Labrador coastlines here. What we can do for each of these strata is estimate in the strata the per capita rates of change across species and summarize those. We can find areas on the shelf that were changing rapidly and changing early. And on this plot, any plot in yellow indicates an area that's declining. Any area shown in blue is an area where abundances are on average increasing. And anything surrounded by a line is one that is statistically significant at a 0.05 level. So where the model thinks there's statistical evidence of substantial change in that period. What we can see is, okay, in the 1980s, populations were definitely declining, but it was limited to a relatively small area of the shelf inshore. But following that, in mid 80s and towards the early 90s, This collapse expanded from that area and covered a much wider region before finally starting to collapse to a smaller area of consistent declines in the 1980s. And then we see in the 2000s a mixture of some areas increasing in blue and some areas declining in yellow. Before finally seeing more consistent patterns of increase around the 2006, and then a switch in the 2010s and 2014s to a decline. But again, we now see a case where the decline is actually starting in the offshore region. So the decline that's happening across biomasses in this case is potentially a different kind of decline than we first saw. So it's something that's starting at the offshore and possibly moving inshore. So in summary, we're able to use these HGAMs to identify not only when did the regime shift happen, but where it happened and how it's spreading across the landscape. And for the last part of my talk, I'm going to touch on a second case study, focusing again more on the spatial aspect of detecting regime shifts. So trying to see what we can learn from looking at regime shifts playing out across a whole wide range of different locations. And so here I'm going to be using HGAMs again to understand the causes of rapid change across landscapes when all you have are limited time series data for a few locations. In this case, I'm going to be looking at walleye in Wisconsin. So we talked about walleye a little bit at the start of the talk. They're a major fish predator, and they're typically found in cool, low clarity lakes. So in general, we think they prefer cooler waters. We think they prefer lower clarity, and they're found spread from northern Canada all the way down to Louisiana in some cases. It's a very important species in Wisconsin. It accounts for a substantial fraction of the $2.7 billion sports fishing industry. And when I started working on walleye for doing my postdoc, and when I was doing my postdoc at the Center for Limnology in Wisconsin, there have been reports of population declines for the last decade. And now we're starting to see cases, more and more cases of population closures where fisheries are being closed for it. And the question was, what is causing this decline? What's causing this transition? And is it only isolated to a few lakes, or is it happening across the board? And one of the things we think is causing this is a collapse of recruitment. So there's many fewer and fewer young walleye. And this is a time series, this is essentially a smooth time series of recruits per kilometer taken from a long time series collected by the Wisconsin Department of Natural Resources from early 1990s to 2014 across a whole range of lakes. So something like 120 lakes, 120 different lakes per year from a collection of about 800 lakes across the landscape. What we see is, if we just look at the raw data, see possibly an increase in recruitment up until 1995, then a slow and constant decline in recruitment from 1995 all the way up to 2015. But we didn't know if this was recruitment, if this was potentially an observation error. So for instance, in the early part of this time series, the focus was on collecting lake data from lakes that were considered to be good walleye lakes, so high walleye recruitment. So was this decline just a sampling effect because we're seeing more lakes that were essentially marginal walleye lakes? And where was this decline occurring? So we wanted to know, when did the decline start? And we wanted to know, where did it happen? So this is a plot showing all the lakes in the state. The Great Lakes are ones that are known to contain walleye at least one time. The blue lakes represent lakes that we have at least one observation of recruitment data from, and the red lakes represent ones that we have at least one observation of recruitment and adult populations from. So we have this huge sample of lakes, a huge population of lakes across the landscape, and only sparse samples of the time series in any given lake. We want to know, how much is this trend consistent across lakes, and how does it vary in space? And one of the things we want to know is, how much does it vary from lake to lake? So this is a time series of essentially 4 different classes of lakes. So naturally recruiting, so ones that the Department of Natural Resources in Wisconsin considers to be naturally recruiting for walleye. Ones they consider to be mixed, where they require at least some stocking to maintain a population. Populations that are stocking reliant, so ones that rely on stock to maintain their abundance, and ones that don't have a known status, that were not classified under this system. And what we see is, we see declining trends in all 4 of these. And we want to know how much does this vary from lake to lake. So each one of all these points here represents a recruitment observation from different lakes, but we don't actually have that many lakes where we have long-term time series data. So each of these points represents an observation recruitment, but it's not necessarily observations of recruitment from long time series. We also want to know what drove these collapses. So were these declines being driven primarily by abiotic changes, so changes due to warming and due to changes in clarity of lakes over time, or is it changes in the biotic community? Because we've seen both shifts. So we look at bass. So bass are thought to be a potential competitor for walleye and are known to potentially prey on young walleye, and maybe preying on young walleye recruits. We know that bass were actually, large amount of bass were actually increasing for a decent portion of the time series and are spatially spread across the lake in varying amounts. We also know that temperature was increasing across these lakes in the same period. We see that temperature was warming at roughly about from 1600 growing degree days at the start of the time series to about 1800 growing degree days above 5 degrees Celsius. And again, we see a large amount of variation, spatial variation in this. So we want to know, are these biotic changes potentially responsible for shifts in recruitment? Or is it changes in abiotic, or is it both features interacting? However, what we had to contend with is we had only very limited information for each lake. So this shows essentially lakes ranked by the amount of data we have. So the 25% of lakes, we have 5 to 7 observations of recruitment. So very limited time series. Another 25% of lakes, we have 10 to 12 points of recruitment. The third quartile is longer time series, and only 25% of our lakes do we have essentially 15 to 20 data points. So we're very data starved when trying to determine how these lakes are changing. And one thing you'll notice from these different example time series is that recruitment is very variable within a lake. So it can be very difficult to try and fit a curve to a single lake and pick out whether its recruitment is declining or not, especially given how sparsely it's sampled from one time point to the next. So to deal with this, we decided to use an HGAM approach, so hierarchical generalized additive model approach. So if we have all these different time series, each for each location, what we can do is actually pull these different time series towards a common trend. So not fit a single trend for everything, but instead fit a separate trend for each one, but penalize those trends towards a common average. So even though there might be a large amount of fluctuations, unless there's substantial evidence for a difference of trend between them, they'll be pulled towards the common trend. Still allows for variations between lakes, but it also takes into account the fact that these lakes might be following common patterns or common drivers across this landscape. So after we've grouped and penalized these recruitment trends towards these common trends, we get this outcome. So each line here represents a trend for a given lake. You'll notice these trends vary hugely between them. But these blue lines represent the common average trend within this class of lakes. And one of the interesting results from this is that we initially had thought that this was likely a case of decline of recruitment among mixed and stocking reliant lakes. These lakes that were doing poorly or were bad walleye lakes would see the brunt of the decline. But what we actually saw was that our naturally recruiting lakes showed the same patterns of decline as all the other lakes. So lakes that we had previously thought, or that the department had previously thought were considered good walleye lakes, showed basically the same rates of decline, although their recruitment levels, their average recruitment, was still higher than the other lake classes. So like in the case of the Newfoundland data, this also lets us look at trying to determine when did this shift actually happen. So from this, we can quantify mean rates of change, so the average rate of change in this trend, as well as variability in our estimates to try and quantify when is a given class of lake declining at a statistically significant rate. What we can see is that basically all of the trend indicates that the lake started declining around 1995 and essentially been staying at a negative declining, relatively constant declining rate since then. So this is again measured on a per capita basis, so essentially measuring the exponential rate of decline for the species. What we see is that basically recruitment is declining essentially exponentially at a relatively small but constantly negative rate from 1990 onward. And that holds for almost all these classes. The only difference being the stocking reliant ones where we have very few data points of samples and we have a harder time getting a reliable estimate of the overall trend. So this lets us identify when the population as a whole, so the whole population of all lakes, or when any given lake started declining, as well as from this we can extract summary statistics like how quickly is the lake changing and what factors correlate with that. So we extracted 2 summary statistics from each lake. The first one being average recruitment across the whole time series, so what the mean number of recruits are, kind of a metric of essentially walleye, essentially lake health, or how good the lake we considered at the start of the time series as a walleye lake. And the second is the slope, so the average exponential rate of decline across the lake across the time series. And then we can correlate these different rates of change with our different factors. What we saw is that based on the expectations, we saw a pretty strong effect of both biotic and abiotic factors on average recruitment. This lines up with what we know about walleye in this system in general, that warmer lakes, we expect to see lower walleye recruitment, and in high bass lakes, as well as interestingly high bluegill lakes, so bluegill being another sunfish, and we think might actually be a predator on young larvae of walleye, even though they're not a major or large fish species themselves. What we can see is that if you look at the average number of bluegill or bass across the lake, across the time series, see a pretty strong effect of this on recruitment. However, if we look at rates of change, we see a much stronger signal of biotic influences, so that we see that rates of change are relatively flat across growing degrees. There's a slight decline in rates of change. So again, negative rates of change, meaning a population is getting worse more quickly. And we can see a relatively constant rate of change for growing degree days. And we in fact see the biggest negative effect from bluegill. This one species that we largely ignored as a potential driver of this seems to be one of the best predictors of which lakes are declining. That lakes that have abundant panfish, or these small species that might be in fact preying on or competing with walleye recruits, are the ones that we're seeing the most substantial declines. Although even in this case, we're not seeing a huge effect size. So this is still only predicting, say 5 to 10, something like 10 to 15% of the total variation in rates of change. So we still don't know the final drivers of these differences in rates of change. So I realize I'm getting around the 45-minute mark. So just to sum up, overall, rapid ecological change is a community and landscape level phenomenon. We can't think of it as just something that happens to a single population, and we can't easily monitor rapid change using a single population or a single metric. If we want to quantify rapid change at community or landscape scales, we need tools like HGAMs that can pool information between groups and detect periods of rapid change across landscapes and quantify change itself. And finally, these HGAMs allow us to pool limited information from a large number of short time series to better understand the causes of change at large spatial and community scales. So just very briefly, where are we going with this work next? What are the next steps of this? One of the things we want to look at is start expanding this HGAM framework to take into account the idea that species are related, have similar relationships based on phylogeny and based on functional traits. So we wanted to see if we can use information for how phylogenetically similar species are and how functionally similar species are to use that information to cluster, to essentially pull species towards each other based on that. We want to say that, okay, species that have similar traits or similar phylogenies might also have similar trends. We might be able to get us more bang for our buck when analyzing these time series. Second, we're working on theoretical models and trying to develop ecosystem indicators to understand essentially how a whole system is changing and trying to determine why cold marine systems seem to be sensitive to these regime shifts. So in this case, we're focusing on size spectrum models, so trying to model the whole spectrum of abundances and communities across time and understanding how these size spectrum models might be, what makes them resilient or not to regime shifts, and how effectively we can detect rapid change in these size spectrum models. So that reaches the end of the talk. Thank you very much for listening. I'll quickly say thank you to just a range of collaborators on this, including QCBS working group that's funded by QCBS and Quebec Center for Biodiversity Science and the Canadian Institute for Ecology and Evolution. well as a large range of collaborators across DFO, Center for Limnology, and now Concordia. So thank you very much for your time, and I'm happy to take any questions.